So on behalf of the Biochemical Society and Portland Press, I am very pleased to welcome you to today's webinar, which is part of our Biochemistry Focus webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. So my name is Cathy Tournier. I'm going to chair this session. I am a reader in the Division of Cancer Sciences at the University of Manchester, so a former colleague of our speaker today. So I lead a research group that is focused on MAP kinase signaling in cancer, and my work centers on understanding the molecular mechanism driving cancer progression and identifying potential therapeutic target. So I have been honored to have been invited to chair this session. So today's webinar hosted in celebration of 120 years of the Biochemical Journal. Founded in 1906, the Biochemical Society's longest running journal has been publishing major biochemical discoveries, starting with a study on tissue oxygen and evolving into a global platform for disseminating modern molecular bioscience. In this webinar, we will hear from one of our past authors, Professor Douglas Kell from the University of Liverpool. Douglas is currently research chair in system biology and director of GenMill. Prior to this, he spent many years at the University of Aberystwyth before moving to the University of Manchester in 2002, where he led the Manchester Centre for Integrative System Biology. From 2008 to 2013, he served as chief executive of the BBSRC, one of the UK's most major public research funding bodies, before joining the University of Liverpool in 2018. Professor Kell is known for co-coining the term metabolome, and recently he has contributed and exploring areas such as antimicrobial resistance, membrane transporters, and ion dysregulation. So more recently, he has contributed to research into fibrin and amyloid microclots and their possible relationship to long COVID and other chronic conditions. I have to tell you that a question will be asked after Douglas's presentation, but you can send in your question during the talk. If you have a question, please type it in the question box as shown in the image on the screen, and we will try to answer as many as time allows. I would now like to hand over to Douglas, who will be sharing his work and findings on the role of Fibrinaloid Microclot complexes in chronic inflammatory disease.
Thanks very much, Cathy and Lucy, and to the Biochemical Society for inviting me to speak to you. As you'll be aware, you can get hold of the recording and indeed the slides after the talk. So I'm going to include a lot of material, some at a high level, and go through quite quickly. And my starting position, except that it's not moving the slide, is that it's a long-term collaboration with Risha Pretorius from the Department of Physiological Sciences in Stellenbosch University, or previously Pretoria. And the opening gambit, as it were, is that long COVID has a massive number of symptoms, over 200, depending upon how you count. There's 28 on that slide. And you might think it implausible that there could be a unitary cause. Here's a variant of the same kind of thing from one of our own studies in the Sankey plot, showing you that all of the things on the left, all of the individuals are having all of these symptoms pretty much. And it also shows you that the female to male ratio of long COVID is in the order of 2 to 3 to 1, which makes it contrast with acute COVID, where more males suffered from acute COVID than from long COVID. So this is the kind of overview of the whole talk, which, as you can see, comes in 4 parts. First of all, I'm going to talk about normal clotting and how it works. Then we're going to look at how blood can clot into an anomalous form and various molecules that will trigger that we've shown experimentally, and the fact that what these anomalous clots are, which we originally called dense matter deposits, is amyloid in nature. And we'll go through what that means in the case of blood clotting. It's obviously not the same thing as the classical amyloidoses, such as A-beta-synuclein and so forth, although these are all diseases with an infectious origin as well. And then finally, we'll look at the fact that the clinical consequences of having these microclots blocking up your system and sloshing around in your bloodstream. And another part of the backstory that was mentioned apropos of the Biochemical Journal is a series of papers that I've had in it. The top one there has had over 90,000 views, so I think that's what persuaded the Society to invite me to give this talk, and we'll go through what they say later. So if we look at the blood clotting cascade, there are two pathways labeled extrinsic and intrinsic. Don't worry about all the details. The key thing is the molecule labeled 2A in yellow, which is thrombin, which is a protease that acts on fibrinogen, which is the second most abundant protein in blood, typically 2 to 4 grams a liter. And what it's going to do is remove two little peptides from the fibrinogen molecule, which is a 9:1 cigar-shaped molecule, as in the cartoon there. And these two little so-called fibrinopeptides, red and blue, labeled FPA and FPB, are then going to allow a remarkable self-organization in which individual fibrinogen molecules line up, bind to each other, ultimately cross-link under the action of another factor. And so there's going to be a fiber, which you see at the bottom, is going from left to right. And the size of that fiber is about 280 molecules per unit length going round and round. So if one molecule is my finger, the whole thing is going to be this sort of size, although we'll see the variation in size a bit. So it is a remarkable kind of self-organization, and what the blood clot is going to look like then, if you look on the left-hand side of these, is essentially like spaghetti. So either using purified fibrinogen in the top left panel or platelet-rich plasma. When you add thrombin, you see this clot that actually just looks like lots of fibers that look like spaghetti, for want of a better phrase. However, in the presence of, in this case, ferric ions, the appearance of the clot is completely different in the electron microscope. And we call these things dense matter deposits. And you can see they're a bit like having parboiled your spaghetti and let it stick together and clag into a rather disgusting mess. So clearly, there was something very different about the blood clots under these circumstances. Now, if you go back to the famous Christian Anfinsen paper in which he denatured ribonuclease with urea and guanidinium, unfolded it completely, removed by dialyzing away the denaturants, and discovered that the ribonuclease would fold into the form that it had before he added the denaturant. And two conclusions were drawn from this. One was correct and one was not. The correct conclusion was that the entire ability to fold up into the tertiary structure of the protein was encoded in the primary sequence, which mostly is true to this day. There are one or two proteins that need chaperones, but for practical purposes, the vast majority, that is true. However, the second conclusion was that this was therefore the most stable form. Now, this would be impossible to know, because if each amino acid has 10 conformations and there are 300, that's 10 to the 300 conformations. So there's no way you could know. You certainly couldn't test. And what was discovered subsequently is, here is a picture on the left of the prion protein, which in its normal PRPC conformation has lots of alpha helices, but as people will know, it can be persuaded to flip into a different, in fact, amyloid form called PRPSC. And indeed, one PRPSC molecule can catalyze the transition of many millions of PRPCs into PRPSC, and that's how prions work. But there's a massive energy barrier separating the form that came off the ribosome, the PRPC, from what is actually the most stable or the more stable form. And that's why normally you don't see the PRPSC, you don't see the amyloid form of proteins, but they are thermodynamically the more stable of the type. So having seen in a variety of circumstances that blood could be persuaded to clot into this anomalous form in which the normal spaghetti-like clots look completely different. We then realized that actually this form might be amyloid in nature, and we'll come shortly to exactly in more detail what that means. And in this paper here is where we showed it by adding tiny amounts of, in fact, bacterial lipopolysaccharide. I mentioned these diseases actually have an infectious origin, so unlike most of the science I do, this was actually a hypothesis-dependent project. So first we looked in the classical way, adding 1 molecule of LPS per 100 million fibrinogen molecules. Now remember, there's no thermodynamic problem. The blood is going to clot. The question is, does it clot into the normal form or the anomalous form? And in the control case, you can see it clots into the normal form, as we saw before, the nice spaghetti-looking thing. But adding this tiny amount of bacterial lipopolysaccharide caused it to flip, in panels B and C, 2 examples, into this anomalous form with dense matted deposits, and also a very different size distribution. You can see that mostly the fibers are smaller, but some of them are, in fact, significantly bigger. The main point is it's completely different. And what we know about amyloids in general is that their defining motif is the so-called cross-beta motif, in which the protein now folds up backwards and forwards, in which the peptide bonds between different chains form linkages to make this cross beta. So it's highly full of beta sheets, different morphologies from the same sequence, and also different sequences will give you different morphologies. And the main point about this is that we can add a molecule called thioflavin T that normally is not fluorescent, because when the photon is absorbed on this single bond between the two halves of it rotates and dissipates the free energy, so there's no fluorescence. Whereas when it's bound to its target, as in the right-hand side here, that rotation is impossible, and so it fluoresces. So the molecule is not fluorescent, but fluorogenic, which means that when you have it in a suspension of clots, the background is nice and black, whereas the clots are going to look bright green. And it's going to place itself along these cross-beta motifs. And that's why it will stain any amyloid, because it's going to stain the cross-beta motifs independent of what the side chains in that particular part of the protein might be. And so here's a picture from that first paper where we added the lipopolysaccharide. The controls have essentially no little green dots, and this minuscule amount of lipopolysaccharide causes this massive flip into the amyloidogenic or the amyloid form. And around this time, we started looking at many other diseases, and we will come to a couple of them, but mostly we're going to talk about non-COVID and ME/CFS. Now, when you look at a fiber of this type, and we'll again go into more of this later, there are 2 ways in which it can interact with other proteins. The first is just by having them kind of stick on the side or be blocked. And actually, that's what it looks like in normal clotting. Many people have done proteomics of normal clots, and to a certain approximation, I'll show a graph later, the molecules other than fibrin, which is the polymerized form of fibrinogen, that are in these fibers essentially reflect the concentration of things in the soluble proteome. But what we will discover is that in these anomalous clots, that isn't true at all. There is a great increase in the amount of particular proteins that happen to be themselves amyloidogenic. We'll go into the evidence for that, and an exclusion of some proteins that are very high in plasma but simply don't go into these forms. And they can go in 2 ways. One, they can be homopolymers which join together to make heteropolymers, or they can be individual heteropolymers where a single strand has multiple proteins within it, and we don't really know yet how that goes, but we hope to find out. At the same time, we also look to see microclots in individuals who just had a stroke. So here's the control again next to no microclots, and here's someone who's just had a stroke, absolutely massive number of microclots that are amyloidogenic. And at this time, Risha was just moving from Pretoria to Stellenbosch. So we never really wrote this up properly, but you'll see that we've done something different instead a bit later. We also looked to see that the lipopolysaccharide could indeed bind to purified fibrinogen using isothermal calorimetry, and of course, as you would expect, it can indeed do so. As mentioned, we found these kinds of phenomena in individuals with a variety of other diseases, all of which are chronic inflammatory diseases and in fact, as I say, have got an infectious origin, which isn't always widely recognized at the time. In addition to thioflavin T, which isn't perfectly specific, it will also bind to DNA in certain circumstances, double-stranded. There are more selective fluorogenic amyloid stains, and in particular these molecules which are conjugated oligothiophene. Thiophene is a little 5-membered ring with a sulfur in, and again, with the single bonds in between each of these things, if you have different chain lengths, they will bind, and the greater the chain length, the more to the red is the fluorescence. So there's a series of these AMI trackers, and as you can see, they all bind relatively similarly in these circumstances. And they're, as far as we understand, much more specific for amyloid, and their chemistry is quite different from thioflavin T. And in particular, there's more evidence for the fact that we've added the bacterial lipopolysaccharide that's caused the flip. If you add lipopolysaccharide binding protein, then you decrease significantly the amount of amyloid that you can see with the AMI trackers. So around this time, we're moving into the COVID era, and we were doing diabetes at the time and comparing it with COVID, and you can see that people, this is acute COVID with acute COVID plasma, absolutely dripping with these microclots. And if you remember in the early days of acute COVID, people talked about a ground glass phenomenon in the lungs of individuals with acute COVID, and the ground glass phenomenon was just our microclots, but that wasn't recognized at the time and maybe still isn't. And the other thing that was different is lots of changes in the platelets at the same time. And again, these microclots, these are very small ones here because that's an erythrocyte, a few microns, are all binding like bilio to the erythrocyte. So the plasma is absolutely dripping with microclots as well. Now, around this time, we were only semi-quantitative. We realized we ought to find some sort of grading system because we were just essentially taking samples because it's hard to get a wide field with a high resolution, although we have methods now. So we used a grading system in which you can see. And I draw your attention to the bottom panel where there's a bright field image here, the amyloid image here, and you can see that pretty much all of the microclots are amyloid in nature in these circumstances. We'll see something similar later. And again, in these individuals with type 2 diabetes, you can see you probably grade them somewhere between 2 and 3, and certainly not 1 or 4. A similar thing was done with the platelets. Again, the stains are for p-selectin, which is the pink, and PAC1, which is green. And again, the platelets in these circumstances are quite different too, although my focus today is going to be on the microclots. So what's happening? Well, what's happening is that the normal forms of fibrinogen, which are these coiled chains, are going to flip into this anomalous form with the beta sheets, which of course are now going to be going not parallel to the large fibers, but backwards and forwards perpendicular to the beta motifs. And so they are understandably quite different. So it's not really very surprising that the larger fibers that you see in the electron microscope are completely different as well. Now, I should say we're not at all the only people who've done this, just for the avoidance of doubt. Clearly, we'll tend to focus on what we've done, but Caroline here and Lara Yonka have both recently published very similar phenomena for folk with long COVID. And not yet published, but doing the rounds with our collaborators is an analysis of the FOSP cohort. These were people who were hospitalized with acute COVID, who either did or did not go on to get long COVID. So that kind of nice inbuilt control. And in this case, the people with long COVID had about twice the number of microclots as the people who didn't get long COVID. Where does it come from? Not bacterial lipopolysaccharide in this case. In the case of the COVID, we showed you can take recombinant spike protein and add it to platelet poor plasma. That's what PPP is. And you can either with or without thrombin increase massively the number of microclots. So again, the spike protein alone, like LPS, is sufficient to cause the flip into the anomalous fibrinaloid form. In other words, we coined that word to be a kind of portmanteau between fibrin and amyloid, partly because the people don't like the word amyloid because it makes them think of classical amyloidosis. anyway, in this case, you can see massive difference between the healthy controls and the COVID-19 folk, whether acute, as in this one and that one, or in long COVID down here. And again, the morphologies are rather different, but the main point is they're all dripping with the green thioflavin T stain induced. Now, we're again not the only people looking at amyloidogenicity. Sophie and Per here in Lund have been long looking at the amyloidogenicity of various things, including in particular parts of the SARS-CoV-2 spike protein chain, and it is highly amyloidogenic. And here they've used Congo red, which is a classical stain for amyloid, which gives this so-called classical apple green birefringence when you look at it under polarizing light conditions. And they also looked at the thioflavin T, and you can see that over a period, the amyloid form is forming and continuing to form over many minutes. Then we're going to start looking at what else is in these clots. And in particular, in this particular case, this was a study again of long COVID, and we then looked at what was in the microclots that we can see of these massive amounts, and again the platelets are there too. And as the title rather gives away, antiplasmin is going to be one of them. So classical proteomics is going to be done here. You just take the plasma, trypsinize it. We had to trypsinize it twice actually, because these amyloid forms, as are well known, are rather resistant to proteolysis. In fact, the test for the prion PRPSC is to add proteinase K, which munches any known protein under the sun except for the PRPSC, and so what's left behind is your test. So in this case, we're looking for things that are increased in the microclots compared with normal plasma from controls, and you can see large fold changes, but in particular, there's this presence of this antiplasmin. So plasmin, which I should have mentioned earlier, is the enzyme that gets rid of the clots normally. So when your blood clots normally, which is clotting and unclotting all the time so that you're ready to go if you have cut your finger, the enzyme that gets rid of it is called plasmin, and it's activated from a plasminogen by limited proteolysis. But there's a molecule that stops that called antiplasmin, and these microclots are full of antiplasmin as well, which also happens to be quite amyloidogenic. Serum amyloid A, surprise, surprise, another amyloidogenic protein, as you'll guess from its name, 17-fold increased in the microclots here. And again, here's the antiplasmin compared with something else. So, in addition to the fact that the clots are normally going to be more resistant because of the fact that they have this amyloid form, the presence of antiplasmin here is going to contribute to making them even more resistant to being proteolyzed or fibrinolyzed, which of course is going to contribute to them sitting around in your bloodstream doing mischief rather than being got rid of in the normal way. And as I mentioned before, there's a phenomenon called cross-seeding, in which the presence of a particular amyloid can cause other amyloids to flip, just as prion proteins in their amyloid form will cause prion proteins to flip. But it doesn't just have to be the homopolymers, the same molecules. It will cause other amyloidogenic proteins to flip. And again, there's a lot more detail in these two papers here, one of which we looked at the amyloidogenicity of proteins in the microclots, another in which we said, well, if that's the case, then the proteins in all these other kinds of clots, such as emboli and atheromas and things like that, should also be full of amyloidogenic proteins, and so it proved. And as I said before, to a significant approximation, the normal clot proteome pretty well reflects the soluble plasma proteome, but ours in the fibrinaloid is completely different and highly enriched for amyloidogenic proteins. So here's an example from a normal clot study where they're looking at the proteins that are present in the normal clots, the ones that look like spaghetti. And here's a paper from Heck and Co in Utrecht, looking at the average protein concentration. And you can see there's a tolerable straight line. And when you do the same thing with our microclot proteome, the correlation is 3 times lower. So really they are very different indeed. Now there's an online protein, or you can get the R code to run it yourself. The online version will let you run up to 50 at a time at the website here, and that's the R code there, but you can find it easily enough. And so what these folk did was to essentially train a random forest on known and not amyloidogenic and non-amyloidogenic proteins to predict sequences that would be amyloidogenic, and over the whole protein it gets a score between 0 and 1. So the question is, how do things shape up? So the first thing we did is to look in UniProt for all the human proteins, well, all the proteins, but this is just the human ones, that were coded by human beings as being amyloid. And in the case of the human one, there were 24, including lysozyme, which is just below 0.7. We'll come to that in a second. But lysozyme is well known. It's used as a model, actually, for looking at amyloid formation. But for lysozyme, you have to move it to pH 3, and then it goes amyloid very easily. So some things. So this is the cause of diabetes. It's called amylin. And massive amyloidogenic score in a myelogram, and that's beta-2 microglobulin. Again, you can see it all in this paper. So we're thinking that anything above 7.7 is likely to be amyloidogenic because all of the known amyloids are. And when you look through all the, you can run a myelogram for every human protein. We did this in that paper. You can see the most common are actually between 0.8 and 0.9. And the massive majority are above 0.7. So most human proteins, and I can tell you just hopefully about to be impressed in the Biochem J, just going through the last refereeing from other taxa as well, are amyloidogenic in nature, which is a kind of biophysical phenomenon. So when we look at now MECFS and ask the question, how many microclots are there, first by area, it's more than 10 times as many in MECFS, which is a disease related to long COVID, not identical, but shares many features, one of which is called post-exertional malaise, which we will come to shortly. So 10 times as much area and 10 times as much intensity, because they're going to accrete and form larger clots, and so there's going to be more intense greenery in each of the spots that are counted in this analysis. We also wanted to check that it wasn't just that you'd had a jab, because if you've just had a jab, it turns out from Caroline Dalton's paper that you're more likely to form amyloid too, which is not very surprising because the jab is either the spike protein or an mRNA capable of encoding it. So, and again, a lot more MECFS papers beginning to figure out that almost certainly the MECFS is well known as a post-viral syndrome, and in these cases, almost certainly herpes viridae that are responsible. We also, with Alan Thierry, discovered that these microclots contain nets, neutrophil extracellular traps, which are particular 73, 74 nucleotide pieces of DNA that are thought to be involved in stopping infections by coating bacteria. And you can see in this case, myeloperoxidase, which is the classical stain for nets. Sometimes it's on its own, sometimes it's with DNA, sometimes it's staining with THT as well, and sometimes it's just THT on its own with no DNA. So you can see very heterogeneous composition of the microclots. Again, it's all in that paper there. Another interesting area, and we'll come to the microcirculation anon, is sepsis, which is, of course, initiated by an infection, but sepsis is, in fact, the response of the body to infection by having a highly overactive immune system responding to it. And this group here, led by Cheng Hock To, who happens to be in Liverpool in the next door building, but a different department, asked the question of people in the intensive care unit with sepsis, of which he had 104, they essentially borrowed, used, copied very accurately our methods for detecting the microclots with thioflavin T, and asked the question: if you've got sepsis and come out dead, are your microclots very different from if you come out alive? And the odds ratio was 5 for mortality and 50 for disseminated intravascular coagulation, which is a very nasty thing connected with sepsis. These are massive, massive ratios. And again, they also did a proteomic study and I analyzed their proteomes. And again, they're highly amyloidogenic proteins in their proteomes. So, you get a paper in the Lancet for a 10 or 20% improvement. This is a factor 500%. These are massive, massive effects. Reacher also acquired a nice imaging flow cytometer. So as it goes through, you can look in the bright field, as here at each microclot, because a clot, we call it flow clotometry, will scatter light and so it can be triggered in the same way that a flow cytometer would normally be triggered with a cell. And you can see, again, each of these is dripping with amyloid as judged by the thioflavin stain in this channel 7. And there's a picture showing the same sort of thing. The controls here next to nothing, and the people with long COVID, massive, massive amounts of stain of the thioflavin T. So I mentioned post-exertional malaise as being something. So one of the classical symptoms of these diseases is fatigue. You try and walk up the stairs and you just can't because you're feeling exhausted. Post-exertional malaise is something different because it is something in which you do some exercise and a day or two later you feel dreadful and referred to as a crash by the poor individuals who have these diseases. And what we think is going on here is a chronic version of what is well known as an acute thing, which is ischemia reperfusion injury. So in ischemia reperfusion injury, which can happen after a heart attack or a stroke or to an organ on between the donor and the recipient getting it, what happens is oxygen drops or runs out. That's the ischemia, and so it's hypoxia, and then it comes back again. That's the reperfusion when it's exposed. But during the ischemic phase, mitochondria are highly over-reduced. So that when the oxygen comes back, instead of reacting with cytochrome oxidase with 4 electrons to make water, the oxygen reacts with complex 3 with 2 electrons to make peroxide and complex 1 with 1 electron to make superoxide. And together these two reactive oxygen species, in the presence of unligated iron, different story, come together to make large amounts of the hydroxyl radical, which is highly toxic to the cells. But what you're doing is reflecting the death of the cells in the 1 to 2 days. And so we think that post-exertional malaise is actually caused by a chronic version of ischemia reperfusion injury, because what's happening is that the microclot's going to be blocking up the circulation, especially the microcirculation, stopping oxygen getting to cells and tissues and mitochondria. So we think that the microclot story can in fact explain self-consistently fatigue in an obvious way. You just stop the oxygen getting to the tissue, so you can't make ATP very efficiently. PEM I've just talked about. Autoantibody production was the third of those Biochemical Journal reviews. If you're a protein that's suddenly been flipped into an amyloid form, instead of being recognized as self, which you would be in the normal form, suddenly you're looking like not self. So again, you have a ready explanation, which hasn't properly been tested, but at least a straightforward explanation of principle as to how you can make autoantibodies when you're flipping proteins into an amyloid form that you wouldn't expect to. POTS is something similar. Again, you try and do something that, in this case, usually it's a tilt table and blood is going to be redistributed in the body. But if the oxygen is being blocked to the tissue, what does the heart do? Well, it speeds up because that's what it tries to do when it feels you're a bit short of oxygen. And again, there's a review on this and there's a review of atrial fibrillation where the original story is the other way around, but we think the microclots cause the fibrillation, not the fibrillation causing the clots. And there's lots of evidence in that review, which I'll leave people to read. The ischemic stroke we're going to see shortly. Now, of course, anything that can block up the microcirculation can cause trouble as well. And this particular very scary paper from the New England Journal by this group looked at the prevalence of microplastics and nanoplastics in various kinds of arterial plaques and so forth. And again, there was this massive hazard ratio of 4.5 to 1 of people who had these things in their plaques. And these are hydrophobic. They will bind to hydrophobic things. The microclots are hydrophobic too. And so microplastics and nanoplastics, definitely not a good thing. And I counsel people to read that paper as well. So, of course, the question then people will ask is, okay, what can we do? Because these things are here, they're not being degraded properly by the normal plasmin that would be degrading them. And Justine, who's the first author of this up here, did these experiments where, again, following Caroline's protocol, we used a microscope which has a good magnification and a small field of view, but it can automate to travel over a large area. So you can get a pretty picture of about 90 megabytes of the microclots as stained with thioflavin T. And in this case, we made our own nattokinase recombinant. You can buy it too, but of course you don't know what else is in there. And what nattokinase is a product of the natto fermentation, which is a soybean fermentation to which is added bacillus. It's called bacillus natto. It's probably a bacillus subtilis strain. And it's fermented to make this rather gloopy thing. I have to say it's quite an acquired taste, but you can also buy the nattokinase because the protease that is doing the work, stupidly called nattokinase, it's a protease, not a kinase, is called nattokinase and is clearly capable of degrading soybeans and other things. And as we will see, it's also capable of degrading microclots. So we're going to make our microclots by adding fibrinogen to thrombin in the lab. We're going to count the number of microclots that stain with thioflavin T, and they're going to go up over time, and they're going to continue to go up. But in the presence of nattokinase at two different concentrations, they're going to come down again because the nattokinase is actually going to work and get rid of them, including the amyloid clots, which was the first time anyone has shown that you could do that. So that's the number against time. And we also look at the intensity over time, and clearly those are going up still because the clots are accreting. We're shaking the tray in these circumstances too to help that along to more mimic the fact that the blood is flowing normally, and so the clots are getting bigger over time as well. And you can look here. Here's the picture of the normal ones, and here's the ones from the nattokinase. Obviously, there's far fewer. And that's just a histogram of the intensity of the green in this case. But clearly, the nattokinase is working here. There are other fibrinolytic proteases that have been proposed to work. One is called serrapeptase. It comes from the silkworm. When the silkworm wants to get out of its silk cocoon, it vomits from its stomach a bacterium called Serratia, not Mark Eston's, but another one, and that secretes a protease that will degrade the silk cocoon, and the silk moth can then fly away. So you can guess if it can degrade silk, it's probably going to be able to degrade microclots. And there's another one called lumbricase that comes from the earthworm. And these are available widely, but you want to get the so-called enteric coated ones so they get through the acid of the stomach. So Martha Ecke here in this PNAS paper studied self-reported things that folk with long COVID had tried, and ME/CFS as well, and which ones made them feel better by varying amounts. And here's the nattokinase. More than half the people are finding a benefit from that, and a whole bunch of other things which I'll leave you to read the paper. But I'm also going to point you at this bottom one here, because there are people around putting it about that there is not a biochemical underpinning to long COVID, but it's all in your head. It's a psychosomatic disease, and you can just think yourself well. And this is extremely harmful, not to say insulting to patients. And people who tried this, both cognitive behavioral therapy and graded exercise therapy, the GET people, more than half got worse. In fact, almost everyone got worse, but half got lots worse. And really, it's just not good that folk are going around saying that long COVID is psychosomatic for their own purposes, because it simply isn't. And I've given you a lot of biochemical markers. And in fact, people are getting so fed up with it that this large number of people wrote to the Royal Society of Psychiatrists to tell them to stop their people doing this. And this is very recent, as you see. Then we got interested in, as I mentioned earlier, with the microclots. The clot if you have an ischemic stroke, one of your main arteries typically in the brain is clogged up with some kind of a clot. And nowadays, these are removed mechanically by sticking a pipe up your thigh, the femoral artery, all the way up to wherever the clot is. And inside the pipe is the thing like a little lacrosse stick, and the clot is pulled out, removed, mechanical thrombectomy, and pulled out, and job done. And normally folk throw these in the bin and don't pay any attention to them, but our collaborator, Alec Kendu here, decided to make a clot bank, and we said, okay, let's have a look to see if these clots are amyloid in nature. And whilst people may think that COVID and long COVID are a kind of marginal thing, which is not at all true, but they don't get the headlines they ought to. Stroke is the second largest cause of disease in the world, and most of them are ischemic strokes. So this is not a small phenomenon. Anyway, we looked at our stroke clots. You can see the size of them here. They're in millimeters now. And every single one, we looked at 8 in the original study, and every single one is absolutely dripping with amyloid, as you can see. Not absolutely everywhere, but large amounts of it, which explains in a straightforward way why they haven't been got rid of. Because you've got to ask the question. Normally we get rid of clots because we're clotting and unclotting all the time, but in the case of stroke, we haven't. There has to be a reason or reasons. And the straightforward one is because they're amyloid in nature and much more resistant to being got rid of. Here's one where we compared 2 of them by looking at the bright field and the staining with thioflavine again. Look at the size of this thing, it's 2 millimeters. And again, this one is absolutely dripping all the way around. This is perhaps more particularly around the edges, but still a lot of amyloid in the stroke clots. Now, the party line is that the stroke clot sort of disappears from somewhere else in the body and then blocks up the biggest artery in your brain. If you think about this, it's not very logical, because it's somehow going to block up the biggest artery, having managed to get through smaller arteries. And I don't think that's a very plausible explanation. What we think must be going on really is that the microclots, which will get through the smaller arteries, can accrete together until they've formed a macroclot, and we're in the throes of checking that out. Again, there's lots of available proteomes of stroke clots as well as the normal clots, and we actually analysed 10 studies in this paper in the Biochemical Journal. Here's just one. The vast majority, again, are having an amylogram score over 0.7 and then indeed over 0.8. So again, these are all fibrinogens are labelled there. So these are all highly amyloidogenic proteins. So it all makes sense as to why you would expect to see amyloid proteins in amyloid clots. So again, just return to this. This is the fibrinogen molecule, and there's a binding site here for the various triggers that we know to date have been able to cause it. We don't yet know precisely how it flips into the amyloid form, but there isn't any doubt that the amyloid form is there because it stains with all these different stains that are more or less specific to amyloid, and the things which they're not specific are different, and yet they all stain in the same place. And indeed, we've shown that fibrin is in there too. So I came upon this idea, and Virchow in the 19th century used the same term, blood stasis, in reading some papers about traditional Chinese medicine. And blood stasis is an absolutely recognized phenomenon. And of course, it's describing exactly what we're talking about. Things blocking up the blood flow and stopping it going in the way you would like, thereby decreasing the amount of oxygen getting to tissues. And so if you look into the literature of blood stasis, you can ask the question, so what do people use to stop the blood stasis? The answer is this cocktail of 11 herbs called shui fu zhu yu, shui meaning blood in Mandarin, and there's the list of things there, but again, you can read the paper. And I have to say, a number of people are reporting very good results, and of course, these cocktails are well known to be beneficial for various kinds of cardiovascular disease, and we're currently looking into trying to find out what the constituents are that may be having the bioeffects and what those bioeffects are. Lastly, just as a slight side note, we've mostly been looking at what I call the structural phenomena, that is to say the presence of the microclots. But what is also interesting is to look at the functional consequences. And here's 3 different kinds of methods that people have used to show the microcirculation not working properly. Nail follicularoscopy and laser-based methods looking at essentially fancy maths on the light that reflects when you shine a laser on a hand or eye even, that show you what is moving and how fast, and that's the only thing is blood cells, and thermal imaging, obviously, if things are not moving as fast as they could. If you dip your hand in cold water, it stays cold a lot longer than if the blood is going through. So anyway, there's reviews on that. So what do we want to do to get rid of them? Well, get rid of the microclots, and which there are a number of ways of doing, but obviously you have to use your physician for this. This is not clinical advice by me. I'm not a clinician. I'm also just going to say, much discussed in the literature, which I haven't had time for today, the literature of long COVID, is the importance of viral persistence. We think that is continuing to explain, at least in part, why long COVID carries on being so long, and endothelialitis. So the endothelium, which is actually the largest organ in the body, all of the cells surrounding your blood system, the microclots can damage those, and they can create the microclots. And anything that has this A causes B and B causes A phenomenon will amplify, and we think they go hand in hand with each other. So again, to revert to the summary slide, we've looked at how normal blood clots into a nice spaghetti-like form. We've looked at things that can cause it to clot into an anomalous form that turns out, which we originally called dense matter deposits, but contains many amyloidogenic proteins, neutrophil NETs, NETs, and other things such as antiplasmin that inhibit. The consequence is that these things are sloshing around your bloodstream and can be seen and measured, and the following consequence, of course, is that they're going to block up microcapillaries and stop oxygen getting to tissues, which at least in principle can explain this vast number of effects that we see in people with ME/CFS and long COVID. And with that, I thank you for your kind attention. I'll just mention this again, it'll be in the slides. I got interested in why people believe bonkers things despite the evidence, and there's some of that in this field and some of the other fields I'm in too. And with that, I thank you for your very kind attention. Cathy is
all right. Thank you so much, Douglas, for sharing with us this I think very important area of research that appear to impact many pathological processes. So now I think, so we have time for questions. I can read some questions that have been posted in the in my box. So I can start with this first one, Douglas. So the question was, among the proposed triggers of fibrinaloid microclot formation, which mechanism appear most important in sustaining chronic disease?
So I think the answer is it depends on the disease. So we know that LPS, and I didn't have time. We've also showed lipoteichoic acid, which is the equivalent cell wall material from gram positives. We've shown unligated iron, which is also iron dysregulation is happening in all these chronic inflammatory diseases, rheumatoid arthritis included, Alzheimer's, Parkinson's, even migraines. They all share the same kinds of phenomena: inflammation, iron dysregulation, and some are clearly post-viral diseases, some are post-bacterial diseases. In the case of rheumatoid, it doesn't seem to be very well known, but the evidence is off the scale clear that it's caused by Proteus mirabilis and Proteus vulgaris. So it depends on the disease, but clearly the origin in long COVID is COVID, and the origin in some other cases is microbacterial.
All right, thank you so much. Now I have a question that is more on the clinical side, I should say. And the question is, since these amyloid clots are structurally different than the typical canonical thrombi and the D-dimer, CRP markers won't work. What type of clinical blood test could GP, primary care health worker use to diagnose them?
Yeah, well, we're hoping that these our methods will become available, whether by because many places have flow cytometers. It turns out that very few have fluorescence microscopes. But of course, it's also got to jump through the hoops. Although, as I say, several groups have shown that they're good markers. People have proposed classical inflammatory markers of various cytokines. Of course, these are not particularly selective for particular subtypes of disease, and if you can cluster symptoms in long COVID, and if you just cluster by symptoms, you find 6 different subtypes. So as it stands, the GP doesn't really have access to any of these tests, and certainly not ones that have been given the nod by the relevant authorities who would allow them. But diagnosing long COVID is, again, I'm not a clinician, but usually it's pretty clear who's got what or should be.
Mm-hmm. So I think your data, with the natokinases and seropeptase, yes, as potential treatment, it looks very convincing. And I think one question was asking about whether there are any clinical trial evidence for these yet, or are we still mainly at the mechanistic observational stage? Because I am sure many of us know people with long COVID, and I don't know whether they are offered that, or do you see what I mean? Where have they been?
Oh, well, no one's offered these things by a doctor, because the NHS doesn't have anything going along these lines. What I would say, and again, it's all in Martha Ecky's stuff. The usual amount of nattokinase that people talk about is like 1 pill, which is 1 to 2,000 of so-called FUs, fibrinogen units, and that is based on the amount of natto a healthy Japanese would eat in one meal. But we're not tending to talk about healthy, thin Japanese. We're talking about unwell, probably less than people. And Martha's data rather imply that you need to go up to something like 8 to 10 times the amount, because essentially it's a seesaw. The clots are being formed and you're trying to get rid of them. And if you don't succeed in getting rid of them quicker than they're being formed, clearly they're not going to work as well as if you do. So again, everyone is different. So there's no magic bullet for sure. But as Martha's paper showed, over half of the people who tried nattokinase found it gave some benefits, and of course that may be a minimum, because many of them, in some cases, they give up, because what happens is that nattokinase destroys the microclots, releasing cytokines, making people feel rubbish, and so they stop it. Whereas those who go on for 2 weeks, again, it's all in the data, then find benefits. So again, just the did it work or didn't it work can be much more nuanced than that as to how well it might have worked.
Did you say, Douglas, that nattokinase is not a kinase? I can't remember.
It's a protease. Yeah, it's a protease.
Oh, it's a protease. Okay. Yeah. All right. Yeah.
It's just a historically ancient name that makes no biochemical sense, but it ain't going to change now.
Now there was in the box, there is an interesting suggestion to you, saying that could the fibrinaloid microclots could have also a physiological function, at least in specific circumstances, to trap bacteria, viruses in the blood, like an emergency clearing system, in which case you would not want to get rid of them.
So that's been one of the proposals as to what A beta does and things like that, the classical amyloidoses, and it's what NETs do. And again, many people have these things, but don't appear to suffer badly from them. So as I mentioned when we were chatting before, there's 25,000 genes. Just give me 10, which come in 2 flavors or alleles of 2-fold variation in activity. So with 10 genes, I can explain 2 to the power 10 variation, 10 out of 25,000. So the fact that everyone is different shouldn't be a surprise. The fact that people are vaguely similar is what should be the surprise. So that's why we think there's not going to be a magic bullet, but because it's not just this. It takes 2 to tango. It's the presence of these things. But if the host is completely resistant, it doesn't matter. If the host is super sensitive, it matters deeply. So what they do normally, doesn't exist, because in the healthy controls before COVID came along, we really didn't see them. So it's not obvious that they do have a normal function. There are cases in which certain amyloids, for instance, in making biofilms, there are bacterial amyloids that are actually valuable for the host in making biofilms and things like this, CSG. But I don't know in the case of the blood clotting into an amyloid form when that's been seen as beneficial.
All right. And I have a question actually, Douglas, about because you mentioned this microclot in the context of infection or disease, but I was wondering whether you have ever looked at the effect of aging on clotting, whether you can see what the probability for this clot to appear increases with age.
We've not found anything that lit up massively. Because, I mean, there may well be a trend on it, but we haven't done huge studies that would be needed to pull out in the presence of all the other things that are going on differently as you age. I mean, the famous Dolan-Pito studies with smoking and health, they actually need to get up to 200,000 people before they could get statistical significance because there were so many confounders that paralleled social class. So bad diet, bad jobs, bad exposure to pollutants, as well as smoking.
Right.
And so there are so many other things going on with aging. We've certainly not done the studies to disentangle that.
Yeah, so the reason I was asking is because obviously when they show the graph on COVID and long COVID, they were looking at the increased risk factor, and there were two. One massive was aging and obesity, and that's why I was wondering whether this is because, you know, you increase the susceptibility for the formation of this microclot.
So that is true of acute COVID. It's not true of long COVID. Long COVID is much more, as I mentioned, women, and we think there are some hormonal reasons for that, but also quite often perimenopausal, and often you will find it's people who've been doing a lot of exercise. So the demographic of long COVID is really quite different from the demographic of acute COVID for reasons that aren't entirely clear. Of course, female immune systems are quite different as well. So we haven't managed to disentangle age as being a major thing from the perspective of microclots.
All right, so thank you very much, Douglas. I think we are coming close to the end of the webinar, so I haven't covered all the questions, but I think you are very welcome to continue the conversation online by following the Biochemical Society on Blue Sky, Instagram, or LinkedIn. I would like to thank everybody for attending, and of course the speaker for delivering this session. As mentioned at the start of the webinar, the Biochemical Journal has been a key forum for sharing research for over 120 years. So if you think that your research delivers clear mechanistic insights supported by careful experimental design, our journal offers a trusted society-led home where your finding can speak with impact to the broad community. I have to declare that I've been working with the Biochemical Journal for many years. It's a fantastic journal. And you also have to bear in mind that all the reviewing and the editing is done by research active PIs. So I think that has a great advantage. So we looked very deeply at the data and make decision on the data that you present. So I would like to encourage everybody to submit, of course. Obviously, moving with the time, the Biochemical Journal also meets all current founder mandate for publishing, whether through our read and publish option or green open access without embargo. You can find out more on our website. We welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. If you have any idea for a webinar in 2026, we invite you to submit a proposal. And you can find more information about the webinar at www.biochemistry.org. All our upcoming webinars are listed on our website, so you can log in. You don't need to be a member. Obviously, we would like you to be a member, but you can access them directly from the website. So It's important to stay connected. The Biochemical Society, being a member of the Biochemical Society, can provide you with this option. And, you know, on a personal level, I have to say I benefited greatly from being a member of the society. So now I would like to thank you all, and goodbye.